Hi, everyone, and welcome to the first seminar of this season. So today I'm really happy to introduce one of my dear friends, Franziska Bergmeier. We met for the first time 5 years ago when we went to a workshop on myofauna at the Azores, and kind of stayed in touch since then. And Franzi, she loves to play with mud and made that to her career and profession. And she is not new here in the US. So she did a postdoc at the University of Alabama a couple of years ago at the Kocot lab. And the workshop back in the day was organized by one of our common supervisors from the LMU in Munich. That's also a university I studied and Franzi studied. And she's also currently working for the Zoological State Collection in Munich. So thanks for coming, and I'm looking forward to your talk.
Yeah, thank you very much, Nina. I'm really excited to be here today and tell you a little bit about the animals that I work with. And I don't just like to play in mud, I also like to play in sand, and I will tell you a little bit more about that. So yeah, I'm really excited to tell a bit about these animals because they're very obscure. A lot of people haven't really heard about them or aren't really familiar with them. So I'm always excited if I have the opportunity to work with them. Okay, maybe I have to use the mouse. Yes, okay. So for this talk, I'm basically going to take you along the journey of my master research, my PhD research, and what I'm currently doing in my postdoc, or what I have been doing in my postdoc. And the animals that I work with are Solenogastres. They are a class of mollusks. So mollusks, everyone knows the enigmatic cephalopods, gastropods, snails, and bivalves, but these guys are usually really unknown, even among zoologists. The deep sea biologist Craig McLean has once called them the fuzzy little mollusks you don't know, which I think is a very nice description because it's very accurate in my opinion. So it's a rather small class of mollusks with currently around 310 described species. In terms of their size, they can be very small, from less than a millimeter up to really large species. They can grow up to 30 centimeters. In contrast to many other mollusks, they don't have a shell, which is why they belong to the superclass of Aplacophora, so they are shell-less mollusks. But instead of shells, their body is covered in these aragonitic scleroids. And you can see this here. Some of these scleroids are kind of like these fine needles that give these animals this sort of like fuzzy-looking appearance. Others are these kind of like, ad-pressed, flatly arranged scales on this very small species right here. Some of these animals mostly live epibentically, so they just use their little ciliary gliding sole that's kind of hidden in this mid-ventral line here on their body, like on their ventral body side. And they use this kind of ciliary gliding sole to just move along the sediment epibentically. Some species, like this really colorful Anamenia here that was described from Japan, live epizoically on cnidarians, so on soft corals and hard corals, and also feed on these animals. We find Solenogastres in all the world oceans, from the really shallow water down to the deep sea, from the tropics to the poles. I basically first got into contact with these animals when I was looking for a topic for my master thesis, and my then later PhD supervisor, who at the time was also still a PhD student, said, hey, I have these like nice little pretty understudied Solenogaster animals that were collected on Bermuda, and this is going to be like a nice little thesis for you. You look at those animals, they were collected at the type locality, so where the species was originally described from, and you can just do like a nice little redescription of those animals with like using modern methodologies. And I thought that sounds really exciting. I want to do this. And the to me, also really exciting thing about this project was that these animals were collected from within the sand. So these are really small animals. You can see this little Solenogaster is maybe like a millimeter in size. So they're really tiny, and they live among the interstices, so like the pores between the sand grains. And I was really excited about this project, and I basically used a lot of traditional characters. I will get a little bit into this in a while, and looked at these animals that were collected in Bermuda at the type locality. And after doing all of this, looking at the traditional characters, I had this huge problem that these characters didn't really fit the original species description. So I had this problem that it wasn't really possible to kind of unambiguously ID these animals, but the characters had these kind of showed just these weird combinations that I also wasn't really able to reliably describe new species. In addition to these problems with the morphological characters, I also had a lot of genetic data for some of these specimens that kind of showed me that I was actually dealing with like some cryptic species in this area. And the genetic data told us that we have like at least 3 species that externally all look the same, but are genetically distinct, and we can't really kind of keep them apart. So we called this a taxonomic Bermuda Triangle because we had this conflict between traditional classification and the genetic data. And that was kind of what really got me intrigued by these animals, because I thought, okay, they are these really exciting animals that seem to be kind of in maybe higher diversity than we know at the moment. And we kind of, need to bring this traditional classification and traditional taxonomy kind of into a modern era. And at the time I thought, how hard can it be? It turned out to be pretty hard. So the first part of my PhD was kind of trying to come up with with kind of like a more like modernized way to approach taxonomy. And the first Solenogastres have been described 150 years ago. And sort of the traditional classification or taxonomy mostly relies on 3 main characters for species identification and delimitation. So the first one is studying the sclerotome, so looking at all the scales and needles that can be found along the body. These can vary from the head to the tail and also look a little bit different if you look, for example, at the ventral side. Then, as many mollusks, these animals also have a rasping tongue, the radula, which can look quite different between different families and sometimes even species. And this is also an important character. And then lastly, and this is where it gets so complicated and why this is also one of the reasons why these animals are comparatively neglected and only little studied, is that for actual genus and species level identification, you need to look at internal anatomy of the anterior and the posterior animal, which you can only do if you section these animals histologically. And that obviously is very time consuming and requires a lot of specialized equipment. And to kind of bring all of this into an integrative modern approach also means, of course, that you would like, or that I would like to include DNA and also look at molecular data. And that can be really challenging if you take into consideration that you have to get all of these characters, section the animal into little bits, and then still find some tissue where you can extract DNA from. So the first part really was trying to come up with some workflows on how to handle this and how to tackle this. And the first one I kind of came up with was specifically designed for very tiny animals like the ones I was studying from Bermuda. And what kind of the approach that we came up with is that first of all, we put them under the scanning electron microscope and kind of document the scleroids. You can see this little guy like turning around here. So I built these little carousels where I would stick the Solenogastres on top of these SEM mounts, put them in the SEM and then just kind of rotate them for 360 degrees, take a lot of photos and try and get like kind of all the scleroids. And this is kind of the really important link to the traditional classification that has been used for 150 years. And then afterwards, once this is kind of documented, I take the animals, extract the DNA, and then we amplify markers from them. And by now we do this with specifically designed markers for the Solenogastres. And this genetic data we can then use for molecular species delineation, which of course is really important if you're dealing with potentially externally cryptic species complexes like in Bermuda. Ideally, you can then use those genetic markers for molecular barcoding. When I started out with my PhD back in 2015, there were only 21 Solenogaster sequences in GenBank, and a lot of them were contaminations, so that at the time really didn't help with IDing the animals. But this has, of course, changed over the years. There's like another little thing that I do when I have multiple specimens of like what I think might be one morphospecies. Sometimes I try to like cut one of the animals in half or a few of them so I can look at the anatomy of the anterior part, and I use the posterior part for DNA extractions, because looking at the kind of anatomy of the anterior part, specifically the radula and the digestive glands, is usually kind of a good, gives us enough information to kind of ID the animals on family level and sometimes even genus level. And then there's the second approach that is kind of useful for animals where I feel confident enough to cut them into 3 sort of parts. And the middle section of the animals is what I usually preserve for DNA extractions, and also to like use the cuticle with the scleroids to look at the scleroid information. The anterior and posterior part get embedded in resin. In this case, I use an epoxy resin. So you can see a part of the animal like in this little block. And then I use like an ultramicrotome to cut this little animal that's embedded in the resin block into lots of thin slices, sometimes depending on like the size of the animal between 0.75 and 1.7 micrometers. And then I just generate a lot of little slides that then get put onto his microscopic slides. These are then digitalized. And I, for example, use sometimes a 3D visualization software like Amira to produce these 3D models of the internal anatomy of the animals, or just like kind of sort of classic, schematic drawings of the internal anatomy. And with this two approaches, I kind of decided to tackle the diversity of Solenogastres that's found in two very different habitats. One of them, the pore spaces of shallow sands, like the animals that were found on Bermuda, so the myofaunal animals that inhabit the mesopsalmon, down to the deep sea trenches. That was kind of the second marine habitat that I was focusing on. And the kind of question is, why those two habitats? So this is sort of like a graph that shows the depth distribution of Solenogastres species. So you can see most of the species have been sort of described from the lower continental shelf, and then very few from the really shallow and hardly any from below 6,000 meters. So when we get into abyssal or even hadal depths, at the time, there weren't any records of Solenogastres. There's obviously a lot of sampling bias involved in this. But these were kind of the two habitats that I wanted to focus on, because it seems like there's obviously a really big gap in knowledge. So why this distinction in 4+6 species here for the really shallow water ones? Well, we have 6 species that are really enigmatic. They're big animals, they're very colorful, and people who kind of, they're sort of easily spotted. But the 4 species that I was kind of, or like, the 4 species where we know so little about are the ones that live in the pore space. So we have 4 species at this time described living within the sand somewhere in the shallow water. And this is kind of what I was focusing on. And so the first aim of this project was to explore the global diversity of these shallow water sand-dwelling species, because we knew from colleagues that they had found them at different localities in the world. So we knew they were definitely out there, but people just hadn't really gone through the taxonomic efforts to really kind of, get down how many species there are. So if we look at type localities of the shallow water interstitial species, again, we see like a lot of sampling bias. These are the 4 type localities for those shallow water species, and they're all around marine research stations that have a really long history in studying myofauna. So they have been the experts who study this habitat, who really collect these animals from this habitat, and all of these species are known exactly from this habitat. So obviously there's like a really strong sampling bias here. And especially interesting for me was, of course, Bermuda, because this is not just where I started out with, animals from my master thesis that were collected there back in the 90s and the early 2000s. But this is also the type locality for some species of the family Myomeniidae. So these are these guys down here. And this family is really interesting because it's the only family of Solenogastres that's only known from the interstitial. So all species within this family only inhabit the pore spaces, so all really tiny and have this very specialized habitat. So that's just from an evolutionary standpoint, really interesting. And again, of course, like from my master thesis, I felt there's still kind of like a score to settle with Bermuda and the animals living there. So for this project, I was really lucky to be working with like a lot of animals that were collected all over the world by a lot of colleagues, and also, for example, Nina and I, when we were on the Azores. So I basically was really lucky to get this almost global sampling from, little animals, my Solenogastres, that were collected from coarse sand and shell gravel at different sampling sites in the Atlantic, Pacific, and also the Caribbean. So basically we had it from different localities around research stations, like in Roscoff, also Bermuda and Florida, but also like from the Pacific and the Caribbean. So this was a really nice sampling. And we kind of used, or I kind of used these first workflows to get a general idea of how many animals we have. And we basically kind of got an idea, or at least an overview of the diversity that we find in all of these samples. And at this point, we have reached like 25 species, but the more we look, the more we find, obviously. And something that I'm really happy about is that we managed to gather enough data to kind of characterize these different families that we find in the interstitial, mostly through morphological data. So we were able to take a lot of nice photos of all of these animals, characterize their sclerites, and for some of these animals, we were also able to supplement that with morphological data or anatomical data. And we actually published this as part of the identification guide to marine myofauna. And we managed to find a way to compile this sort of into a taxonomic key that only focuses on external morphology. So this makes me really happy because it kind of makes it possible for people to at least get an idea of the animals that they find if they look for them, without having to go through this super complicated process of, cutting these animals into different sections and studying their internal anatomy. So for anyone who's interested at least in maybe getting a gist of what sort of Solenogastres they might find in the samples, this is like a really comparatively easy way to get there. So at the moment, this project is still ongoing, and yeah, it's been ongoing for a while, but I hope we can soon kind of put it to an end and maybe publish the results we have at this moment. So we're still working on this together with Megan, who at the time was a PhD student in Kevin Kocot's lab. She's now a postdoc at the University of Copenhagen. And I'm just going to show you a little tree in a minute that kind of compiles 105 Solenogastres species from 10 families. And includes 55 interstitial specimens. This is just like a little maximum likelihood analysis based on 2 mitochondrial markers, so 16S and CO1, which are kind of our standard barcoding markers. Don't worry about not being able to see anything, because I will point out the really interesting kind of like, neat facts or neat results we find in this tree. And the first thing I kind of want to point out that even though we're only using 2 mitochondrial markers, the topology we get in this tree is very similar to large-scale phylogenomic studies by Kevin Kocot or also by Megan, which was just published last year. So I think this is actually really exciting. So the first thing we see in this tree, so highlighted, are the clades with representatives, or like the clades that live interstitially, is that we have multiple independent pathways into the interstitial habitat. This isn't really surprising, but it's still kind of neat to see it in the tree. What's really exciting also, I think, and kind of curious, is if you look at sort of the first split of Solenogastres. So this is a large clade that is formed by some interstitial species over here, and then this group over here, which is representatives of the family Amphimeniidae. And this is in so far really interesting, as these are very kind of opposing morphology. So Amphimeniidae are really, really big animals, and the Myomeniidae are these really tiny guys that live in the sand. So that's actually really interesting to see that this is sort of the first split among Solenogastres. And this is also something that was kind of reflected in Megan's large phylogenomic study that she published last year, where she ran 2 different analyses based on transcriptomes. So in the maximum likelihood analysis, the Myomeniidae, or the tiny interstitial Solenogastres, were always retrieved as sort of sister group to all other Solenogastres. But when she ran Bayesian inference, it was always the Amphimeniidae, so the large scale, or like the large sized animals that were sister group to all other Solenogastres. And that's, again, really interesting because it implies 2 very different scenarios about the evolution of body size. We also have some first emerging distribution patterns in these tiny sand-dwelling guys. For example, in this clade down here, which all seem to belong to the family Lepidomeniidae. So this is like a large clade with specimens of sand-dwelling Lepidomeniidae that were basically collected all around the world. But we have this one species down here, or this one clade that most likely is one species that we find in different localities in the Caribbean and also in the Florida Keys. So this is kind of really exciting because it's the first time that we can sort of, based on genetic data, show some distribution patterns. And again, like thinking about how these sort of really tiny animals that live in the sand probably don't lay a lot of eggs, and we don't really know what kind of larvae they have or how long the larvae stays in the water column. This is also really interesting from a point of view thinking about how do they distribute. And then, of course, the family Myomeniidae that I'm so interested in. In this tree, we have several specimens that externally all seem to belong to this family, but we end up with them in 3 different positions in the tree. So this, kind of really implies that we need to do some revisions over here and kind of maybe take a closer look at them. But what's also, again, kind of nice is that there's like a clear sort of geographic separation according to these clades. So we have one clade over here that's collected in the Caribbean and Belize and Panama. We have this one clade of Myomenid-like looking animals that's collected from Washington, close to Friday Harbor Lab. And then we have what we call the true Myomeniidae because they were collected all the way out here in Bermuda in the early 2000s. So our next step is to really kind of look closer at also internal morphology and see if we can find any really nice characters that would help us to kind of delineate these 3 clades and maybe, establish them as like their own separate things. The Bermuda data set is again really interesting because in these animals that we collected on Bermuda, we actually ended up finding 4 genetic lineages, which kind of makes the whole Bermuda mess a little bit more messy. So at this point, we think the taxonomic Bermuda triangle is actually not a triangle in Bermuda, but it's a square with 4 distinct lineages, at least genetically, that we kind of cannot differentiate morphologically. And for me and my PhD supervisor, that was enough reason to say, hey, we're going to go to Bermuda and we have to collect those animals ourselves. So actually 3 days ago, I just returned from a 2-week sampling trip to Bermuda to solve this taxonomic Bermuda square. Our idea was that we would spend 2 weeks snorkeling around beautiful beaches like this, just collect sands and then sort through it in our little lab. Fix those specimens for genetic and morphological data, and then hopefully solve this taxonomic Bermuda square. So we had a little Airbnb rented out. We brought our own dissecting scopes. We had a little microphotography station with us, and then we just ended up collecting a lot of sands. And as you can already see on this photo, so we did end up going to over 16 sampling sites and kind of snorkel in sort of shallow to lower intertidal water. The weather wasn't always very nice. It was actually most of the time, it was really cold. It was freezing with the wind chill. We had temperatures of 45 Fahrenheit. So it was really cold to go snorkeling. It wasn't exactly the kind of tropical field work trip we had envisioned, but it was still really exciting. And we were really lucky to be able to go there. You can see like the waves crashing outside on like the outer rocks. So a lot of sites that we wanted to go to weren't even accessible to us. We thought we'd be smart and go in early December when Bermuda is maybe a little cheaper than during the summertime, but that really kind of made sampling a little bit more of a challenge. So we went to a lot of really nice exposed sites also out on the reef, but we also collected sand right in the boating channel that's like in front of the marine station on Bermuda, because we knew from colleagues that they had collected those animals here before. And this is kind of what it looked like. So we would put on our wetsuits, put on our down jackets, wear hats, carry a lot of buckets, go snorkeling. This is my supervisor where she's trying to save the sand bucket with our precious sample. And we're getting back onto the beach through the waves. On Bermuda, as a tourist, you cannot rent a car. So we use public transport. That meant that sometimes we would miss the last bus and we had to walk, but that's okay. That's all part of the fun when you do field work. And overall, this trip was still really successful to us, because after kind of like having a little bit of difficulties in the start, we actually ended up finding overall 15 specimens of these tiny animals that we were looking for. And we think that in our samples at this point, morphologically, we can distinguish 3 species. So these are probably 2 of the classic Myomeniid species, and this is probably a representative of Lepidoptera. So these 2 are pretty small. This one's kind of like a larger sized animal. And what's kind of exciting about this one, if you look at this photo of its head, you can see, maybe you can kind of faintly see this little cirri, like these stereocilia in the front, that are kind of probably sort of some sort of sensory organ. And you can maybe also very faintly see these little pits here. We've never seen this before, and this has also never been described before. So we're kind of really excited about this. I think we might have seen this on specimens from Antarctica that we observed for a little bit, but this has never been described before. And we're really kind of excited and curious to see what we have right here. So of course, we didn't just find Solenogastres when we looked at the myofauna. We also found some other cool things, and I'm just very briefly going to show this to you because maybe you will never see this again. So these are just kind of 2 very wormy looking guys, and these are actually gastropods. So I think that's pretty cool. You can see they're perfectly adapted to living like among the sand grains. On first glance, this to me just looks like a flatworm, and it was for a really long time described as a flatworm, until someone figured out that they actually have little gastropod larvae, because they don't have a radula. So like the typical feature that would always allow a mollusk to be identified as a mollusk is missing in this animal. You can also see it has these little scleroids. So I think that's a really peculiar looking gastropod. Same with this one. It just looks like maybe a nematode or something. Sometimes it moves a little bit like a flatworm, but this is Helminthope psamobionta, which was also described from Bermuda. And this is a new species of the genus Rhodope, which has not been reported before. So we're really excited about this. We also found some really interesting hydrozoans. This question mark doesn't mean that this is something excitingly new. It just means that I haven't gotten around to trying to ID it. So these are like little hydrozoans that just kind of crawl through the pore spaces and live in there. We also found this Eleutheria dichotoma, which is called the crawling medusa. So it's super tiny. This is maybe 2 millimeter in diameter. You can just kind of see it crawling through the sands. You can see it's budding right here and here. And then something that we're also we're really excited about, which we'd never seen before, is this one, Siphonohydra adriatica. So you can see the little hydroids are attached to like sediment or pieces of sand. And then they have like these 2 tentacle crowns. And this was once described from the Adriatic and the Mediterranean. So these are just kind of neat little finds that we made along the way. And at the same location as all of these hydrozoans, we also found one of these Lepidomeni in Solenogastres. And we know that a lot of Solenogastres feed on hydrozoans. So my PhD supervisor, as a good supervisor, she said, you should put them in the same Petri dish and see, if anything happens. Of course, I was very doubtful, but we actually placed both of them in the same Petri dish and just waited a little bit. And then we could see the animal, the Solenogaster, crawling up on it. And here, he's actually he's ingesting an entire tentacle, which is why he looks kind of so big in the front. And that's just kind of the confirmation for us that this little animal is apparently feeding on these hydrozoans that live at the same station. And I think that's kind of pretty neat, like a pretty neat find, because usually it's really difficult to figure out in these tiny animals what they actually eat. And for this, we also took a little video. So this is very shaky. It's me with my smartphone through the eyepiece of the camera. You can see him lifting up his head, and then you can see kind of how he's opening up this area in the front of his head, which is most likely like a sensory pit. It has some sensory organs in it. So it's like this huge area, and he's kind of almost sniffing around. And now this was after feeding. So he has already ingested the entire tentacle. Now he's just going to move away. And for us, this was a really neat observation, because we always thought, oh, they must be feeding on these animals. But we had gut content sequences, but we didn't really have a picture to put with those sequences. So for us, this was actually really exciting. So we found 3 morphospecies. Next up is checking the molecules. So I will put a lot of them under the scanning electron microscope, and afterwards look at their DNA and see if we can kind of maybe solve this taxonomic Bermuda square. So if we look back at sort of this distribution, like the step distribution of Solenogaster species, we can at this point add at least 25 more species at the in the shallow water interstitial. And it's such a specialized habitat. And again, it just really shows that there's like a really strong sampling bias. And when people start looking for them, people will start finding them in most places. And now kind of like off into the deep. So there are like 30 Solenogasters at that point that were described from abyssal depth, so down to 6,000 meters. And basically from hadal depths, which is kind of anything that you find in oceanic trenches, there weren't any species reported. And I was really lucky that for my PhD, I had this data set of animals that were collected in the Northwest Pacific off the coast of Russia and Japan. There was a series of German-Russian deep sea expedition. This is a collaboration that had been going on for a really long time. With the invasion of Ukraine, obviously this has stopped and is not continued, which is very sad for various reasons. But it basically allowed me to work with animals that were collected outside on the open abyssal plain of the Northwest Pacific, in the sort of semi-isolated Sea of Okhotsk and the abyssal basin that's found here. One of the abyssal straits that connects this basin with the open Northwest Pacific. And lastly, also the Kuril-Kamchatka Trench, so the slopes in the bottom of this trench. And in 2015, I was lucky enough to join one of those expeditions. I was part of the cruise that went to the Sea of Okhotsk. This is me, my friends Rachel and Kirill from Moscow. And this is a photo of the main sampling gear that we used for my animals, which is a so-called epibenthic sledge that is kind of designed to collect epibenthic macrofauna. And over here is kind of like an image of the diversity of these animals that I found in the Northwest Pacific. So you can already see they can be really colorful. They look really different. These photos are not to scale. So animals like this one are really small. They're maybe like 700 micrometers, while like this one is one of the really big representatives. But you can already see that they're just like kind of a really beautiful diversity. So to work up kind of this material, I looked at a lot of scleroids under the SEM. I also did like a lot of different light microscopic imaging. And yeah, I just tried to document as much of the scleroid diversity that we find in these animals. I also picked out some lineages that were really interesting to us and used them kind of for like serial sectioning. So I studied histology. We looked at this 3D reconstruction. So down here, you can see the nervous system in green. This is like the cerebral ganglion in the front. Over here is like an entire animal with still its cuticle attached. Once this disappears, green is nervous system. These are all glands in the head. Blue is digestive system, and everything red are gonads or related to the reproductive system. So this is a little species from the Kuril-Kamchatka Trench, which we described. And lastly, I, of course, did a lot of DNA extractions of these animals. And with this combined data set, it was possible, based on genetic data, again, 16S and CO1, so 2 mitochondrial genes, to delineate 60 candidate species in this data set. There were an additional 13 species where it was impossible to get molecular data. But considering that prior to this work, we had only 11 species reported from the Northwest Pacific, this was like a really nice improvement. And based on all the scleroids, which again are kind of like the link to this traditional classification system, it was possible to ID 80% of the species, at least on family level, and 50% of species on genus level. This was kind of also the first phylogenetic study on family level, because for several families we had multiple representatives, and it kind of confirmed what really did not come as a surprise to anyone who's ever tried working with these animals, that a lot of these groups, these families, for example, the family Dondersidae, are polyphyletic, same with the family Probotinidae, and that, yeah, the current classification system really needs to be revised. Another question that's kind of interesting when you get a data set that's kind of, spans different adjacent regions is like if you compare these regions. In this case, the abyssal basin in the Sea of Kocot, that's kind of semi-isolated, the open abyssal plain out here in the Northwest Pacific, and then, of course, the trench, because this is so deep and it's such an interesting, again, specialized habitat. To kind of really very roughly break it down, we found comparatively low diversity in the Sea of Okhotsk, but with like higher abundance, which might be related to some anoxic events back in the days when a lot of bottom-dwelling fauna was wiped out during last glacial maxima. Whereas in the open Northwest Pacific, we find a comparatively high diversity of species with very low species number, individual numbers. And of course, really interesting to me was especially the data or the animals that we collected at the bottom of the trench, because there had been no reports of Solenogastres living in these depths in our ocean. And so I was actually really happy when we ended up with 11 species that were collected here almost down to 10,000 meters, and also not in very low individual numbers. So we overall had 11 species and 24 specimen numbers. And this is kind of like, yeah, sort of like, of course, it's interesting to see how much the fauna overlaps between these regions based on this kind of limited data set that we have at the time. The big problem is that 2/3 of the species we found were collected as singletons. So we only found one single animal representing those species. And this is kind of plotted over here. So this is the number of singletons plotted against like the depth where they are found. And you can see that like we found a lot of singleton out on the abyssal plain. But what's really interesting here in this regard are these two species, because they were kind of like broader distributed in some sense. So the first one is Cropomenia genslere, which a master student of mine later described. And this is actually a species that we found at several stations in the Sea of Okhotsk, but we also found it out here on the open abyssal plain, one single individual. So this is a haplotype network based on 16S again. I mean, if this is like one population, I don't know, or if this is just a poor little Solenogaster that was one day drifted out and is now like this lost loner. I don't know, it might be possible, but at least for us, it was a nice find. And then there's this species. So this is a still undescribed species of the family Acanthomeneidae. And what's really interesting here is the depth range across which we find this animal, because we collected it at the bottom of the trench at almost 10,000, like 9,600 meters, and also in the abyssal basin of the Sea of Okhotsk, which give it kind of a depth range of 6,000 meters, which is quite a lot for an invertebrate. So if we go back to this graphic from the beginning and look at kind of like the species data that we can add to this, then we have another 47 species that we collected in abyssal depths and another 11 species that we found in hadal depths. And this again just really underlines that obviously there's like a really strong sampling bias in this data set. A couple of years ago, I got invited, so actually this past year in 2023 and 2022, to join 2 research cruises to the Japan Trench, which I was really excited about, because the Japan Trench is kind of it's kind of the extension of the Kuril-Kamchatka Trench that I'd been working with during my PhD. And I had this idea of, hey, wherever we look for these animals, we find them. So this will be really interesting. It was it was like a 3-week cruise for benthic sampling. So really kind of ideal for getting more samples together with colleagues from the Atmosphere and Ocean Research Institute at the University of Tokyo. So we spent a lot of time at sea. We spent a lot of time collecting samples and sorting through those samples. But in the end, I only got 12 specimens, which to me was the confirmation that you don't necessarily always find them where you look for them. So at this point, for me, it will be interesting to think about why we found so little Solenogastres in the Japan Trench. Along the same line, I got invited to join another cruise on the German research vessel Meteor this past October, November. This is a really interesting area because it went out to sample along the Weimar fracture zone and the mid-Atlantic ridge. And back in 2014, there was the Weimar transit on the precursor boat, which basically used the same sampling gear in the same region. On this cruise this past October, we were kind of up here, so very close to this data set that was collected 10 years ago here. And during this cruise, they found 180 specimens of Solenogastres and several morpho species over here. So I was really excited to get back there and kind of collect a new data set that would be suitable for molecular studies. We overall did 60 box corer deployments. We did 24 epibentic sledge deployments, trying to get as much samples as possible. And we spent a lot of time sieving the sediment on deck and spent a lot of time live sorting because it's very warm. But if you work with animals that are collected at like 5,000, 6,000 meters, they're used to cold water. So we had to sort on ice the entire time, sometimes also working in the cold room. And after 35 days of night shifts and very little sleep, I ended up with 7 specimens. So that again was like, to me, really interesting. And now the question is, why are there so few in this area when 10 years ago they found hundreds of them? So this is kind of really interesting. And then like another little project that I would just kind of like to quickly touch upon because I think it's really kind of interesting is this little project that a student of mine did. With Solenogastres, there's always this hypothesis that they mostly feed on cnidarians. We find them like living on corals, on soft corals, hard corals. So this must be like their main food source. We also have this phenomenon that whenever we try to amplify nuclear genes, like the histone 3 or 28S, which are also like typical barcoding genes, we always have this problem that we end up with contaminations from the gut contents because the Solenogastre genes have weird secondary structures that make it really difficult to get these genes with regular PCR, and then we often just get the gut contents. And for a long time, we thought that's so annoying. There's nothing we can do with this data. But then we had this this master student, Lucas, who basically took my entire data set collected in the Northwest Pacific. So he took the DNA extracts and just used kind of generic 28S primers to amplify and just see what he can get out of that. And it was really interesting because he found out that apart from cnidarian, which still like a lot of these animal feeds on, some of the families feed on, for example, terribilids. We have some species that feed on bivalves, some that feed on monostyliferan nemateian. So it kind of showed us that the food source diversity of Solenogastres is a lot broader than we previously thought. And it's also really interesting because 90% of the Solenogastres species seem to only feed on one single food source. And we basically only had one species that seemed to prey on different organisms. So we were able to sequence different individuals of these species and got different prey organisms back. So this was like a really neat little project that kind of came from something that we thought was a really annoying lab problem. So to kind of sum it up, kind of like the research of the past couple of years. So this integrative taxonomy, of course, is like a really efficient way to characterize the diversity, which then in hindsight or like looking forward is the foundation for any kind of biodiversity research. We have a lot of new candidate species in both of these investigated marine habitats. And we're also finding some really interesting first distribution patterns. We're getting a little bit of insights into the biology of deep sea Solenogastres and what they feed on, and now also kind of on the interstitial ones. And we also kind of have the confirmation that obviously we need to revise the current classification. So something that I've been working on with my colleagues from the Kocot lab, Carmen, she was at the time a postdoc at the Kocot lab. She's now at the Smithsonian in Washington, DC. And we kind of started this online platform. It's called APLAC-based, and it kind of provides like a lot of information on these little Solenogastres that we care so much about. We have basically compiled applicable literature that's out there. We're working on little identification guides that are kind of like easy to access. And yeah, we have a lot of data to put out here. Like we want to put out a lot of photos so that it just makes it easier for people who might potentially be interested in trying to ID these guys to make it a little bit more accessible. So you've heard me talk a lot about discovering new species, but I've said very little about describing new species. This is like the classic conundrum in biodiversity research, I guess, that discovery rates are so quick and so fast and description rates lag behind a lot. I calculated that once if I were to describe all the species I found in my PhD and in the past couple of years, it would take me about 9 years. I would enjoy that, but I think it would be really difficult to get funding for that. So I think it kind of, at least in Solenogastre taxonomy, it's sort of time to think about different taxonomic approaches that can sort of differentiate between deep taxonomy, where we do the full workflow, and kind of rapid approaches that just sort of have the idea of getting the data out there. And that's something that I'm working on right now, and also colleagues from Kevin Kocot's lab. And for me, this project is kind of like funded by the Ocean Census from the Nippon Foundation. They gave me a little bit of a grant money to do that, which I'm really grateful for. And then, of course, we're currently also working on getting a stable phylogeny with mitochondrial genomes that we can kind of use as a backbone to finally revise some of these families that really urgently need revisions. And then lastly, towards integrative Solenogastres systematics, we're not a lot of us working with these animals, but those of us who do are really passionate about them. There's Kevin Kocot's lab with his grad students, Emily and Chandler, who are both like doing really fabulous work. I think Emily is working on a really gigantic barcoding data set, which is going to make all of this a lot easier in the future. And Chandler is just doing great taxonomic work. Same with Carmen, who's now at the Smithsonian. She's publishing one species description after the other. So there's really a lot of exciting stuff happening in Solenogastres research. And with that, I'm at the end of my presentation. Thank you very much for your attention. There are a lot of